Okay, so in the last one we had a look at the structure and functions of different kind of immunoglobulin isotypes produced by B cells. In this one we're going to take a deep look at the B cells themselves, how they mature, how they're selected, how they're activated, where all this is going on, and we're going to talk a bit about how they actually choose what kind of immunoglobulin isotype they're going to produce. So let's get into it. B cell activation. B cells develop in the bone marrow. So remember from one of the earlier videos we talked about the hematopoietic stem cells being the original pluripotent stem cell for all immune cell and blood cell lines. The hematopoietic stem cell can differentiate into one of three different types of progenitor cell. It can either become a myeloid stem cell, which produces the cells of the innate immune system and platelets. It can become a erythroid stem cell or an erythroblast, which will produce your red blood cells, or they can differentiate into lymphoid stem cells, which are our B cells, T cells, and natural killer cells. So they develop in the bone marrow after they've differentiated from these lymphoid stem cells. And mature B cells then have a membrane-bound antibody on their cell surface. And for these mature, as in like they've finished developing, they've not finished their like maturation process to become plasma cells. So when they're still inactivated, but these B cells will either have the antibody IgM or IgD on their cell surface. In order to produce different immunoglobulin isotypes, they'll need to go through a process called CD4-CD40L interactions that we're going to talk about in a minute. So once activated, so once these B cells come into contact with an antigen that their immunoglobulin variable region recognizes at the antigen binding site, the B cells become plasma cells, shown at the bottom right here. And these plasma cells secrete antibodies. Activation is triggered by antigen binding on membrane-bound antibodies on these mature B cells. It can either be T cell mediated, so these B cells can become activated and differentiate into plasma cells by a T cell bringing an antigen to the antibodies on its cell membrane, or it can just pick up an antigen that's free floating. If a T cell presents it an antigen on its T cell receptor, then we say it's T cell mediated maturation, and if it is picked up free floating in the body's tissues or extracellular fluid or blood, then we say it's T cell independent. And this kind of T cell dependency or independency is important to what kind of isotypes it can actually produce. The immunoglobulin isotype produced by the plasma cell depends on whether activation is T cell mediated, like we just said. Follicular helper T cells are the antigen presenting cells to these maturing B cells. They transform B cells into either short-lived plasmablasts or long-lived plasma cells, or memory B cells. The follicular helper T cell has special surface molecules that direct the B cells they direct the follicular helper T cell to the germinal center of the lymph nodes, where they're able to interact and present antigens to these naive kind of mature B cells. The CD40 to CD40L interaction is responsible for B cell isotype switching. Follicular helper T cells can do this upon binding to a B cell as the B cell secretes follicular helper T cells can only cause a plasma cell to change isotype of its immunoglobulins by binding to that B cell. So only when the T cell receptor presenting the antigen binds to the immunoglobulins on the B cell, can the B cell differentiate into isotypes? Because isotype switching is also dependent on co-stimulatory molecules produced by the B cell itself, which are triggered by this antigen presentation on T cell receptor. So the germinal centers of lymph nodes, shown in this image on the right, with these kind of blue, light blue, green circles within the darker green circles. You can see it if you look carefully, the label in the middle top one. The late stages of follicular helper T cell mediated cell differentiation occur within these germinal centers. And going into a bit depth about this interaction, basically the CD40L receptor on follicular helper T cells presents an antigen and interacts with the CD40 receptor on B cells, which causes the isotype switching of the immunoglobulin molecules. These follicular helper T cells are activated only upon presentation of antigens to these on these receptors that we talked about, the CD40L, CD40. Upon activation, the follicular helper T cells secrete cytokines that induce isotype switching in the B cell. And the type of cytokine secreted by the follicular helper T cell is going to determine the isotype of the ultimate plasma cell that the B cell is going to turn into. But in order for this process to occur, you need chemical signals within the B cell that we talked about here, the co-stimulatory molecules. Activated B cells undergo this isotype switching as well as a process called affinity maturation, which is very important. The same thing happens with developing T cells within the thymus to make sure that their T cell receptor has a high affinity for the antigen that it's going to pick up, usually presented on dendritic cells. So with regards to B cells, this affinity maturation is going to make sure that they're producing antibodies with antigen binding sites that are more specific to the antigen. So they're going to have a higher affinity to the antigen on their antigen binding site and form a stronger bond. The B cell clones with the highest affinity to the antigens will proliferate, while those with a lower affinity are going to undergo apoptosis because they're worse at their job. They have they form weaker associations with the antigen, so they're not as useful. So we get rid of them, and we proliferate the ones with higher affinity. The immunoglobulin isotype is dependent on the type of antigen as well as these cytokines produced by the follicular helper T cells, and anatomic site of infection is also important. For protein antigens, we require follicular helper T cell involvement with B cell differentiation to initiate isotype switching to go from the ITM, ITA immunoglobulin isotypes that the naive mature B cells have. We want isotype switching to IgG immunoglobulin isotypes, which take faster effect and have higher affinity for antigens. These target the protein antigens, but for polysaccharide and lipid antigens, they don't require follicular helper T cell involvement, because to deal with these polysaccharides and lipids, they're important for dealing with capsulated viruses, so bacteria with a lipopolysaccharide capsule. We mostly opsonize these with IgM immunoglobulins. So, like we said before, the naive mature B cells can either produce IgM or IgA already, so they don't need this CD40, CD40L isotype switching to produce the type of immunoglobulins they need to deal with that. So these B cells don't have to worry about needing T cells for their maturation. And finally, quickly on monoclonal antibodies, they all have the same variable region as they all come from clones of a single B cell. We'll go more into